scientists in France and Canada have built a tool that can flip a switch inside brain cells and boost their energy production. When they used it in mice with dementia-like symptoms, the animals regained lost memories. That's a striking result, because it suggests that low energy inside brain cells may not just be a side effect of diseases like Alzheimer's. It may be one of the causes. The team focused on mitochondria, the little power plants inside cells. Neurons demand huge amounts of energy to keep memory circuits running, and previous studies have often found weak mitochondrial activity in people with neurodegenerative disease. Until now, though, it wasn't clear if those energy problems were driving the memory loss or just showing up after the fact. To get at the answer, the researchers created a special receptor called mitochondrial. It was engineered to sit directly inside mitochondria and respond only to a lab-made drug. When switched on, it cranked up energy production inside the organelle. Tests in cells showed stronger mitochondrial function. Then, in mice bred to model Alzheimer's and frontotemporal dementia, activating the receptor in the hippocampus, the brain's memory hub, restored their ability to recognize new objects. That's important because the improvements happened even though the hallmark disease features, like protein buildup in the brain, were still present. It indicates that part of the memory loss may come from fixable energy deficits rather than irreversible brain damage. The same tool even protected mice from memory problems caused by THC, the psychoactive compound in cannabis, which is known to dampen mitochondrial function. The study doesn't mean people with dementia are about to get this treatment. The technique relies on gene therapy and direct injections into the brain, which aren't currently feasible in patients. But it does provide evidence that mitochondria are not just passengers in neurodegenerative disease. They may be active drivers of decline, and that opens up new directions for therapy. More work will be needed to see if the same approach can work in human brain tissue, and if safer ways can be found to target mitochondria. Still, this research marks a turning point in how scientists think about energy and memory in the brain. You can find more details about this study and others like it at scipost.org.